Any questions? Yeah. No, no, just the stuff that's been, just the feedback that you got. It could be like fix this issue or elaborate on that or fix the APA style or something like that, right? So hopefully it's not really a fundamental change that you have to do. Um, I imagine it's just there's a degree to it. It depends on what submission you made initially. But you don't, yeah, you probably don't want to redo the whole thing. Just take the feedback and then fix the stuff that were pointed out. Question?
What's the next assignment or quiz?
Well, the next this week we'll talk about more about visual pathways. Next week we'll talk about object recognition, and then the following week we'll have an exam. Let's see, where did I? So today is the 10th. Yes. So in 2 weeks we have the second exam. Um, this resubmission is not a big deal. You just do it quickly, and then you'll focus on your exam two, which is in 2 weeks. So we'll have like next Friday or something like that. There will be an exam study session, just like we did it for exam one. We only have one other assignment really, which is after exam two, you'll have this presentation assignment, which you'll present the paper, record yourself, and then you'll submit the recording. So there's only two assignments.
Isn't there a final paper?
Does that under the umbrella of assignments? Yes.
Oh, yeah.
Later on, you'll submit your topic, um, and then you'll submit your final paper. Yeah. Questions so far about? I mean, you done one exam, what is one assignment? The semester is practically over now. But meaning that you've gotten a hang of the course. It's not really, we have such a straightforward process, and like I don't think there is anything confusing. But if there is, you should let me know. Okay, so last time I kind of was short on time, so I kind of glossed over a few things. I'm just gonna elaborate a little bit more, and then we'll go to the new stuff. So what we were really talking about last time were these visual pathways that are taking visual information from the eye, and then one pathway that we did not elaborate on takes that visual information, goes to a midbrain area called superior colliculus. But we did spend a lot of time talk about the other pathway, which taking visual information from the eye and bringing them to primary visual cortex. These two are low-level visual pathways. Low level because they're really close to the sensory modality, and they're more primitive. Like even, some of these pathways, even our long ancestors, like even, lizards, like have some of those visual pathways. So they're low-level visual pathways. Today, we'll spend a lot more time on high-level visual pathways, pathways that are in the cortex. Now, when I say pathway, what are you imagining? What do I mean by a pathway? Yeah, so neurons that are connected in one layer to neurons in the next layer, and then those neurons are connected to the next layer. So one layer has a ton of neurons, like retina. Retina is like a two-dimensional representation of the visual world. And that is basically a sheet of neurons, right? These are a sheet of photoreceptors that are connected to ganglion cells. And then those axon fibers are collectively moving and basically connecting to an area like LGN, And then those neurons, and these are new neurons, right? They're not the same neurons. They just received input from the eye. These neurons have their own behavior, and then their axon goes to the primary visual cortex. So when we talk about pathway, we're talking about different areas that are connected. Each area consists of neurons, and these neurons are connected. So they basically send their axons to the neurons in the next layer. Those receive information, they process it, then they send it on to the next layer. So that's what I mean when I say there's a pathway where information that is at one stage creates a neural activity few stages down, few layers down. You kind of, this is like a feed forward process. That's what it's called. And I'll elaborate on that term a little bit more later. So when we talk about law of a pathway, we said, okay, the last stop for that pathway is the primary visual cortex. And primary visual cortex is some people call V1, V1 part of V2, like different people may use different terminology, but when we talk about cortex, again, I glossed over a little bit. So as I said, the cortex is really a sheet. It's not the shape of it because you want to fit it within a skull, it bunches up. But in reality, it's a sheet. And I saw a demo somewhere that I thought communicates this point well. If this plays on my, so imagine this is the cortex, right? Like it's a very simple demo, but I think it communicates the point I'm trying to make. That this is the cortex, and then when you spread it out, it's gonna become a flat sheet. But when you let go of it and basically try to fit it within a skull, it bunches up. Again, this is not exactly the shape of a cortex, but think of it that way. So if this is what it looks like, it means that you could spread it out technically. And there are different ways to spread it out. Just made a few slides here. These are repeat of some of the other slides. So how would you spread it out? Well, you could say, if I take the cortex and basically put some air in it, I could blow it up like a balloon, right? So it becomes, each hemisphere becomes this blown up version of itself where you could see all the things that were bunched up. Alternatively, you could actually open it up as a flat surface and talk about how each patch on this surface is a brain area. So when people talk about different cortical areas, they're technically talking about a patch on a two-dimensional surface. Does that make sense to everyone? So that's what, so when we talk about V1 in reality is that, okay, it's on the cortex, you open it up, it's a little strip at the bottom is V1, for example. Then V2, V4, different brain areas. Yes. No, so these are different animals, like humans and macaques. That's why it looks a little different. But depth, so depth in this, so because it's a cortical surface, right? And it has a little depth, like few millimeters. You could talk about depth in that sense. You could also talk about depth in terms of processing. So if visual input comes to V1, goes to V2, and then we'll talk about goes through these two pathways that we'll talk about, then you could talk about processing depth, which is, okay, how many levels of the cortical layers or cortical processing it has gone through. Depth, what do you, does that answer your question? This one? Okay.
So when I was looking at the blue one on the bottom right, I was just trying to figure out or looking at going deeper.
So this is a flat surface. But it's a volumetric with a flat surface. So let's say it's a complex looking balloon. So it has a volume to it, but you know, the surface itself is flat. It's not really like the cortex itself is like this, right? So it's not a flat surface. It's flat if you look at it under a microscope, the cells are all on the surface and they just basically go in and out because as I said, it's like a bunched up. Um, now if you put air in here and like open this up, like I should, like open this up, then it becomes a volume but has a flat surface, something like that. So different brain areas are technically themselves flat patches. So V1 is a flat patch that when, you know, it's creased up, it goes inside and just outside. But in reality, it's just a flat patch. And the experiments that we saw, for example, they would put an electrode, like insert an electrode right here or right here on the on like basically perpendicular to that surface and try to record from it. Again, these are different ways to display this. So V1 would be a part of this surface. V2, V3, and all the other brain, like cortical brain areas would be patches on this flat surface. Now, there are a lot of brain areas that are not on the cortex, right? We have a lot of subcortical areas. You know, you read about amygdala, for example, or we talked about LGN last time, which is in the thalamus, or you know, there are hypothalamus. There's, you know, there are a lot of different subcortical brain areas like hippocampus that you heard of that are not part of cortex. But when we talk about the brain area in the cortex, we're really talking about a flat patch of cells, and they're connected, right? So if the cortex has this shape, all the white matter are connecting. Those are the connections, the axons that we talked about, that are connecting these cells to each other. I just wanted to elaborate on that. I hope it helps you build a better mental model of what, when we talk about the cortex and it's a flat surface, what that means. It really means that. So we talked about how now that surface itself has a little bit of depth. So it's not like just single layer of cells, it's multiple layers of cells that are on top of each other. And I described it as if there are, you know, multiple dinner napkins that are on top of each other. And the projection that we talked about, the projection that comes from the LGN to the primary visual cortex, it really projects to a single layer here. This, let's say this whole thing is like 3 millimeters, like really small. But then it projects to a certain layer. There are different cell types that are in V1. We talked about symbol cells, and they have receptive fields like this, that they respond to elongated, basically patterns of light, which is at a certain orientation. We talked about how these cells get their selectivity by combining responses or simpler cells. Now the idea of hierarchy again comes back here. Think of the brain as a pathway, and the pathway has hierarchy to it, meaning that each layer can build on the representation of prior layer. And that's how you get more and more complex representation or selectivity as you go up the hierarchy. Yeah, we talked about these before. These slides are here just to give you an idea of where you find these cells in the brain. Okay, let's move on to part 2. So we talked about simple cells. In the visual cortex, primary visual cortex, you also find complex cells. These are the naming they use then. Now, a complex cell in this context is a cell that is tuned both for orientation, but also the direction of motion. So it only responds when there's a certain direction of motion. So if you have, so the orientation really matters. So if you have a certain orientation of light, and it moves in one direction versus another direction, you get a much bigger response. Now, at this point, you should all know what this means. Each line, each vertical line is an action potential. So a schematic like that is showing that for certain pattern of behavior, for a pattern of light, the cell is responding much strongly. Its firing rate is much higher because you see a lot more action potential next to each other. So yeah, so complex cells have this property uh, where they not only are responsive to the orientation, but also to the direction of motion. And in the video that I showed, we didn't get to that part. They also show the behavior of complex cells. If we have time, I could bring that up again so you'll see in the actual behavior of a cell. You could hear it's firing based on those patterns. Now you could imagine a cell could get that selectivity. How? By combining responses of simple cells. So if it's only responding to simple cells that are at a certain location in a space with certain orientation, it could potentially have a certain selectivity. We're not going to go, it's just a schematic for you again to see that this selectivity is built in this hierarchy, layer by layer. Okay, so what does the primary visual cortex do? Well, as we talked about it, if it is responsive to edges, if it has these center surround antagonistic receptive field, it means that it responds to contrast. That's what it responds to. If a cell responds when there is light at certain area and there's no light next to it, right? We talked about it. The highest kind of firing rate you get from those cells is that contrast, when there's light in the excitatory area but not in the inhibitory area. So these cells can really respond to detecting edges. So if there's an edge somewhere in this space where the contrast changes, these cells respond the highest. So before we get there, let's talk about the organization of V1. So V1 is a large area, and it's the first area in the brain that receives visual input from the LGN. An interesting property of it is that it has so-called cortical magnification, which means that a relatively large area of V1 is devoted to analyzing information from a relatively small foveal region of the retina. Now, we talked about how special the fovea is. And another thing that makes fovea really special is that a large part of V1 is really focused on the information that comes directly from the fovea. So when you look at something, and that's when the light from that thing basically reflects onto your fovea, that's your fixation point. A large part of the V1 is really devoted to processing that information. And that's what we call cortical magnification. The other property of V1 is what we call the columnar organization of V1. This only makes sense if you understood the concept of it being a surface. Because columns, so this is a schematic. Imagine this is the entire surface, entire depth of this cortical sheet, right? So it means that from top to bottom of this thing is about few millimeters. Now you could talk about columns here. Each of these, basically all the neurons that are connected to each other that go across the depth would be one column. Now the V1 has this columnar organization, meaning that each of these columns, they share properties. When the neurons are in one column, they share certain properties, and people have mapped out different columns. Let's look at some of them. Now, this work is all from the experiments that you've seen and the video that I showed. They were the ones that really get that deep into trying to understand what is it that V1 do? So let's look at this figure. So again, this is the cortical surface. This is the entire surface of this is the entire depth of it. We're saying that there is something called a location column. This is a patch of the V1, patch of cells, that is dedicated to processing information from a specific area in the image. Patch next to it is processing information from area right next to it in the visual world. And what is that called? What is that property called? Yeah. So this is the retinotopic mapping, or an example of topographic mapping. When you look at patch of the cortex, patch of V1, and this patch is processing certain visual input from a specific area of the world, a patch next to it is processing a area next to that one. A patch next to it is processing an area next to that one. So this is the retinotopic mapping. But what these people found, the first thing is that, well, there is this retinotopic mapping. That is specific, a small patch of the cortex, this is patch of the tissue, is representing visual input from a specific area of the visual world. So each column, and this is a big column, is coding for a different region of the visual space. So the cells within this column are all responding to the same visual world. It means that if you move the light around, all of these cells will be responding to the same visual part of the world. They have the same receptive field. So the idea, the receptive field is correct, is that all of them have the same area of the visual world. So they all have the same receptive field. The fact that a patch next to it, a column next to it, has a receptive field is slightly adjusted. That's what retinotopic mapping is. So now within each location column, there are two ocular dominance columns. So not only you have this retinotopic mapping, you have that the visual world that comes from a certain place from the two different eyes comes to the same location in the cortex. Does that make sense? So, but notice that these are coming from two eyes, right? So what does that mean? That means that as I'm standing here and there is a light over there, the light falls on my left eye, falls on the right eye. Which part of my left eye would it fall on? The left eye. If it's coming from that part of the world, the temporal side, right? So there are axon fibers from the temporal side. It's gonna fall on the nasal side of my right eye, and these two gonna go, and so okay, which one of them has to cross over? Which one? The left one? So if the left one cross over, which hemisphere would be processing that? The right, but the visual input is coming from the right side. If the visual input coming from the right side, which part of the brain should that end up at? The left side. So which one of these visual input has to cross over? The nasal side of the right eye has to cross over. So because the visual input is coming from that way, it falls on the nasal side of the right eye, it has to cross over to the left side. The left side, the temporal side of the left eye will stay on the left side. What is that crossover point called? Optic chiasm, all right? So the nasal part of the right eye, the axon fibers, will cross over to the left side. Now, what's interesting is that when these two reach the LGN, then they project into the visual cortex in the back, they end up next to each other. Does that make sense? That's fascinating, right? The fact that that it's organized that way, that they end up next to each other on the left hemisphere in the left visual cortex. Now, why would you want that? Why would you want to have input from the two eyes next to each other? What does that get you? The whole image? Depth perception, right? So if you have a slight, so what, how do you get a lot of depth perception is between the two eyes. It's the small discrepancy, right? If I'm looking at an object, I'm looking with different eyes, right? If you close one eye, look at it, close the other eye, look at it, it has a slight difference. And that's what gives you a lot of the depth perception. So it's important to maintain information from the two eyes separate as you go into the visual cortex, because then you want to use that discrepancy between the two eyes in order to perceive that. All right, so within each ocular dominance column, there are these smaller columns, which are called mini columns. And these mini columns are orientation selective. So as we saw, different cells are responsive to different orientation of light. But the cell next to it is responsive to a slightly modified orientation. Cell next to it a little more. Cell next to it a little bit more. So what the brain is doing here, it's really tiling this space with all the possible feature detectors. So if there is an edge in part of the world, you want to be able to detect it. So you want to have all the possible edge detectors for any possible location from either eye. Does that make sense? This is so, but okay, but I was gonna show you this figure. It's not like this is just a schematic, you know, it's not like exactly a square like that. When you look at orientation selectivity in the brain, it looks something like this. So they're next to each other and they have a more continuous process, you know, like cells become selective to one orientation and slowly to another orientation and then another orientation. So it's much more smooth, these transitions. It's not the way we're showing it here. It looks like it's such a clear cut between, you know, different columns and whatnot, but that's not reality. The reality is that it's much smoother, the way you go from one selectivity to another selectivity. There are terms that I use a lot and I use them interchangeably, right? I say these cells have this selectivity, or they're detecting this, or they're responding to that, or that, right? All of those things mean the same thing. What does it mean? When I say the cell has a certain selectivity or detecting something or responding to something. No, so what does that mean for a cell's behavior? When I say, well, this cell is responsive to this orientation, or this cell is detecting this orientation. What does that mean? Exactly. All of those terms mean that this cell that is on this part of, so this is a tissue of cortex. So this is specific cell down here fires most when it sees a horizontal bar in its receptive field. Now, when we see that behavior in a cell, we could just paraphrase it as saying, well, the cell is detecting that because, well, it's responding when that thing is in its receptive field. The cell is, very responsive to that. The cell is like that. The cell is tuned for that, right? All of those things mean the same thing. It means that is when the cell is at its highest activation, firing rate. It means that this is what really fires up the cell. That's what all those expressions means. That's why it's called orientation tuning in V1, because you're saying, well, this cell or this column, mini column, is tuned for that orientation. So it's kind of responds to that orientation.
Share an example of like what the orientation of the object is. Is it like when something is facing the sun and you see a glare on it?
No, orientation really here means orientation, right? So the orientation of this thing means like how angled it is.
Okay,
So in here, like this thing has a certain orientation.
yes,
That's all I'm saying. That's all that means. So, but the world is a complex place, right? You open your eyes. I have these edges. I have all sorts of edges in front of me. But the cell, given now that you all know what retinotopic mapping is, if I'm standing in front of this monitor, this area of the visual world would be processed by some cells in my left visual hemisphere, right? So the cell that is really looking at this area, and now that you know it's a retinotopic, it means that there is a column for every possible location, and that cell, those ones that respond to this orientation would be most active.
okay, so for example, with the little microphone
Yeah.
shifted that way, there are certain cells that are like more tuned to perceive things that are at that angle of slant when I see them,
Yeah.
and the others are looking at more like perpendicular things. Like,
Yeah, but they wouldn't be as responsive in this scenario, right?
yes,
So cells, so again, think of it, when you open your eye, right? Every every part of the visual world is being processed by some patch of visual cortex. Does that make sense to everyone? Like you open your eye, this the light that comes to your eyes end up in your visual cortex. And every part of this visual world is being processed by some patch of the visual cortex. As long as it's in your field of view, it has to happen. Now, if it's going to certain patch of the cortex, well, what's the activity going to be? Well, it depends on what the visual world is giving you. If it's all horizontal bars or vertical bars and horizontal bars in my visual field, those are the cells going to be most active. Does that make sense? So every part of the visual world is being processed in parallel by my primary visual cortex. And cells that find what they like, they're going to respond more, and cells that don't, they're going to be as quieter. That's what that means. Yeah. Sure, that's where it starts. It starts in the retina, then it moves to the LGN, then it moves to the primary visual cortex. And then it's being processed in the primary visual cortex. So the organization of the visual cortex, you mean? Yes, right. So as we discussed it last week, the projection to the projection to the eye, right? So if this is the image that is out there in the world, you get a slightly different image to each eye. So the right eye looks at this image and it flips them horizontally and vertically, and the left eye also flips them horizontally and vertically. And that's the organization, right? It's we call it retinotopic organization because the retina has that organization and the rest of the brain just follows that organization. So the world is represented in this way, and then as we talked about it, it just that organization is maintained in the rest of the brain. So when I say your primary visual cortex is processing the visual world in parallel, imagine if you're looking at this picture of Isaac Newton. What happens is that this is your right hemisphere. So when you look at the brain, so this is section right down the middle, and then you're looking at the right hemisphere. If you're looking at the right hemisphere from that angle, basically, this is where V1 is. And the left side of the visual world projects there. Now, it doesn't project this way, right? It goes through a lot of processing. But it's just to tell you that a tissue in this part of the brain is processing visual input coming from this part of the world. That's what it meant to tell you. That's what I mean when I say you open your eye and your visual cortex is processing the entirety of the visual world. No, the flip is happening in the retina. The flip, because the retina has a lens, it flips the visual information horizontally and vertically. The flip starts in the retina and is maintained across the brain. So yeah, so this figure, as I said, shows you that different parts of the visual world are processed by different part of the primary visual cortex. Clear for everyone?
Question on actual fMRIs, do you ever see like an image as it's
So I have a figure here. Yeah, yeah. So it depends on what you're subtracting. Now, if it is, do I have a figure here, right? So this is a schematic, right? So if there is nothing else in the world, and you have just, again, brain is generally very active. So you have to have a baseline where you subtract from. So if you have a baseline, like a resting state activation, and then you show this visual input, and then you subtract them, you could get something like this appearing in the fMRI machine, where you could see the ring kind of showing up in the behavior, in the neural activity. Yeah, this is like an fMRI. And people have been able to, but as I said, they have shown it with letters too, but you have to have the right kind of baseline because fMRI doesn't look like this. It's like all of it is post-processed in order for you to get figures like that. Is that clear to everyone? Yeah? All right. So what we're talking about here is really important, meaning that the entire visual world, yeah, this is so important. The entire visual world is projecting to your retina, and that visual input is projecting to your primary visual cortex. So in a way, you have a retinotopic map of the world in your visual cortex, where every part of the visual world is being processed. Like as I open my eyes, right? And again, later on, we get to the concept of attention and that the fact that you cannot process everything to the same level. But in the primary visual cortex, you're processing everything almost to the same level. Later on, it becomes harder and you have to attend to different stuff. But in the early visual areas, you basically are processing the entire world. Question?
Did you say there was cortical magnification in
Yeah.
V1? Would that not be like more attention to like foveal vision?
Yes. So that is part of what makes, as I said, fovea special. And we have examples here, for example, right? Here you see the cortical magnification, right? The eye, which is very, so this is the point of fixation where my mouse is. You could see the eye is magnified significantly, right? And the rest, the hair and all these areas are kind of squished. You see that? Do you all see that? Here, another schematic of it, that these areas that are close to each other are going to expand out. You see, so if this is the fixation point, you see the distance between F and 1, it really magnifies on the cortex, on the surface of the cortex. The distance between 2 and 3 is much larger, but it becomes smaller. That's what cortical magnification is. It means that the areas that are close to the fovea get a lot of tissue assigned to them. The same you see here. The areas that are close to the fovea get a lot of tissue assigned to them. And that, yes, that creates a certain this imbalance between information coming from the fovea and information coming from the periphery, which we'll talk about more later. But that's, yeah, that creates all informations aren't treated equal. The ones that are coming from your fixation point are given more tissue in the cortex. So that's the cortical magnification. So Any questions about this? You all get what I'm saying? Other than the fact that there is cortical magnification, we kind of assume that all of this information is being processed. Process is a dubious term. I don't know. When I say it's being processed, what does that mean? When I say the information, the visual information from this part of the world is being processed in this part of the tissue, what does that mean? Okay, it becomes neural activity, another dubious term, representing that part of the world. So representing it in a different format. If I want to capture all the visual information in that part of the world, and I have certain orientations, you could assume that this tissue this part of the tissue that has a receptive field there would have cells that are that are tuned for certain orientation be more active than cells that are tuned for other orientation. That's what representing that visual world in this tissue means. Does that make sense? So you want to maintain that visual information in neural activity. And if the visual information is certain oriented bars, then you want to represent it by those cells that are tuned to those oriented bars. And the cells that are not tuned would not be as active. Hopefully that's clear. So this is where V1 is in terms of its processing. We talked about V1 has that simple and complex cells, but what it really worried about are those orientations. Now, you've seen pictures of the brain so far, a bunch of them. So you kind of, when you look at this, you could tell that this is the back of the brain. This is the front. This is the temporal lobe. Up here is the parietal lobe. Occipital lobe would be back here. Kind of, I wanted you to know where the lobes are. Now, V1 is back here as we talked about it. Like in all these figures, when we talk about V1, primary visual cortex is back here. From there are two cortical visual pathways. Now, these are not subcortical anymore. These are cortical. These are entirely in cortex. And a pathway is a sequence of brain areas. That's what a pathway is. And when I say it's an entirely cortical pathway, it means that all the brain areas are in the cortex. That's what that means. From V1, information can go in two directions, visual information. It could move temporally, right, in a pathway that is called ventral pathway, or you could move toward parietal lobe, and they would call that dorsal pathway. So these are the two pathways, and these are you could call them high-level visual pathways in contrast to the low-level pathway that we talked about, or you could call them cortical visual pathways.
You said ventral, temporal, and ventral pathway,
or dorsal pathway, visual information go toward the parietal lobe. So people have given names to these pathways based on their functionality. People argue that the ventral pathway that goes from the occipital lobe to temporal lobe is like the what pathway. This pathway is for complex pattern analysis, for object recognition, for face recognition, for those sorts of things. The dorsal pathway, they argue, is for spatial processing, for where objects are, localizing objects, those sorts of things. And a lot of evidence for these things came from, and again, later on confirmed by fMRI and whatnot, but a lot of them came from lesion studies. So let's say you have two monkeys, and in one of them, you're lesioning the ventral pathway, which basically means that you're lesioning the temporal lobe. In one of them, you're lesioning the parietal lobe, which is lesioning the dorsal pathway. Now you have them do an object discrimination task. Like, look at these two objects, are they different or not? Something like that. And then you could see that a monkey that had damage to the ventral pathway, what people call the what pathway, could not do this task. And the monkeys that had damage to the parietal lobe, which is the dorsal pathway or where pathway, could do this task. So with this evidence, people were like, okay, there must be some dissociation between the functionality of these two pathways or the areas that are in these pathways. And that is that one of them is involved in object recognition. One of them is involved in recognizing patterns, and the other one is not. So you get a single dissociation between what and where at least. Now, alternatively, if you had a task where the rewarded location was a space was the one that was more spatially proximate to a landmark. Let's say this is the task, or the one that was farther from the landmark. You basically put the landmark, and then you make sure that the food is always on the other on this side, which is closer to the landmark. Then you could see which monkey can do this task, and then people have observed that the monkey could do this task when they had damage to the ventral pathway, but they could not do it when they damaged the dorsal pathway. So this gives you information that, well, maybe there is this double dissociation that is modular, that the brain is processing a spatial information through the dorsal pathway, and it's processing the object information through the ventral pathway. So that's when the whole what and where came from. A lot of it was from these lesion studies. Now you all are kind of familiar with what lesion studies are. You know, we went through them and we saw an example of it. You know, the kind of limitations they would have in terms of trying to, you know, dissociate different roles of objects. But this is a lot of cognitive neuroscience in a way, right? A lot of cognitive neuroscience is to try to assign a job to different brain areas. A lot of it is try to give a functionality or functional specialization to a certain brain area or to a certain neuron, or to a certain, you know, patch of neurons, right? That's sort of a lot of cognitive neuroscience. So this is a prime example of trying to say, well, this pathway or this set of brain areas are specialized for this task, and this set of brain areas are specialized in this task. Now, a lot of people argue after the initial, a lot of people argued after the initial, maybe let's take a break. Let's take a break, 10 minutes, and then we'll come back to it. Okay, so as we talked about it, a lot of cognitive neuroscience is for this process of trying to figure out what different brain areas do or different visual pathways do. So as you could imagine, if you want to assign such a role, a functional role to a pathway, you would, you know, people would debate this. People would be like, okay, is this really the role that it has? Is it really processing the what or the category of the object, or is it processing something else? How about the dorsal pathway? Is it really just processing the spatial location or a spatial relationship between objects, or is it, you know, something else? So an alternative or somewhat newer way of looking at the dorsal pathway is people argued maybe it's not really about a spatial relationship, but rather it's about how we interact with the objects. It's like it kind of takes us closer to how do we want to interact and act upon objects. So the evidence for that came also from a lesion-based study. is a patient DF who suffered damage to the temporal lobe. So the temporal lobe would mean that it's damage to the ventral pathway. And the person had been suffering of visual form agnosia. So visual form agnosia is something like this. You give the person an image, like a model of an apple or a book, and then you ask them to copy it. Just the image is in front of them, they're supposed to copy it. And as you could see, this person who has damage to the temporal lobe is not able to do that. But when you ask them to draw it from memory, draw an apple from memory, draw a book from memory, they get much closer to what you would imagine an apple or a book would look like. This type of agnosia is called apperceptive agnosia, where a person cannot even copy. Not only they cannot recognize an image as being an apple or a book when they look at it, they cannot even copy an image. Like if you give them an image, they cannot copy it the right way. And this is all damage to the temporal lobe. This is the damage to the part of the ventral pathway, the pathway that connects the occipital lobe to the temporal lobe. Now, alternatively, if the patient was asked to look at an orientation of a bar and tell us what that orientation is, it couldn't do it. Not alternatively, but similar to that. Now, when you look at, this is too fine grained. So when they were faced with this sort of, this is a picture from the paper itself. When they were faced with this, with the control, basically what the orientation is, and they were told to, okay, can you tell me what that orientation is? They were not able to do that. But if you put a slot in front of them and they were supposed to take this panel and put it through that slot, they could do it. So that's very puzzling, right? You could imagine when a person cannot look at this, when they look at this slot in front of them and you ask them, what is the orientation? They cannot do it. But if you give them, tell them, put your hand through it, or give them a panel, a flat, paper, and then tell them, put it through it, they could do it. So that's very puzzling, right? It tells you that yes, if they cannot tell you what the orientation is, they might have a problem in terms of recognizing this thing, recognizing what the orientation is. But they don't have a problem in terms of interacting, like acting upon it. So the orientation matching, which is basically requires you to recognize what the initial orientation was, the person fails. But when you ask them to place the slot in there, they could do it. So they took this as evidence that, well, that's true that the ventral pathway is dedicated to feature analysis because it has to analyze what the orientation is. And the dorsal pathway is really devoted to controlling the action. And because this person did not have any damage to their dorsal pathway, they were able to do this task. So this story most likely is somewhere in between, right? That the dorsal pathway does a bunch of different stuff. The ventral pathway does a different bunch of stuff. And these experiments are really probing a specific aspect of that. And they're lesion studies, so all of them have to be taken with a grain of salt, like how much lesion there is, what's the source, like all of those things, so you have to consider all of those things. So these aren't like clear-cut evidence per se. So putting all that together, let's talk about the entire visual pathway, right? From the retina to the cortical pathways. As we talked about it, the visual input hits the retina, right? This is the first stage. That's where the transduction happens, where the light becomes neural activity. This goes to the lateral geniculate nucleus, which is a midbrain area, and that goes to V1. And this area, right, we call that the area V1, V2 would be the primary visual cortex. That's what we call primary visual cortex. And these are all part of the occipital cortex. So occipital cortex is much bigger. Primary visual cortex is part of that. And the way what V1, V2 do, a lot of it has to do with motion, form, color. These information are segregated. What V1 does, all right, this is jumps ahead, sorry. So what V1 does, as we talked about it, and again, this schematic is supposed to show that you have selectivity to orientations in V1 and V2 to some extent, where you respond to the orientation of bar. This is very different than what we saw in retina, where the selectivity was really to circles, right? Those circular center surround receptive field really are detecting circles of light in this space. Those next to each other could become selectivity for you to respond to oriented bars. Now, the other stuff also are in the occipital cortex. So if V1, V2 are responding to oriented bars, motion and color and other information are also processed in here. Processed in here means that there are cells that are most active for a specific color or for a specific direction of motion. That's what it means. So, for example, damage to the area of V4, and V4 does a lot more than this. But one issue that happens when you have damage to V4 is color blindness or achromatopsia. Now, interestingly, people who have damage to one side of their V4, like the V4 in one hemisphere, and this would be damage to which hemisphere V4? Which hemisphere? Left, right? If the V4 area in your left hemisphere is damaged, you're going to be colorblind to the visual world on the right side, because that information has to be processed there. So just to give you a mental model again, do I have a figure here? When we talk about these areas, the areas that I have schematically there, again, these are patches on the surface of the cortex. So the information, the visual information reaches V1, and that information is reaching V2, V3, and then it goes in this direction, and also it goes in this direction. V4 would be something down here. Again, this is a schematic is going to simplify a lot of these things. But each of these areas that I'm mentioning is practically a patch on the surface of the cortex. So V4 would be some area here that you could damage, that you could basically lesion. Or some people have a stroke or something, or have a seizure, and that area are basically in an operation, they just cut it out. Yeah. 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 This would be what they perceive. Yeah. It's not, it's not like something that it's actually like this and they can fill it in. No, this is what they perceive. They see, like when they open their eye, like this part of the world is black and white, kind of. So this sort of thing where you have damage to one part of the brain, one side of the brain before that causes achromatopsia would be hemi-achromatopsia, right? Half. Like hemifield or, hemisphere or something like, just think of it that way. It's like half of something. So there are other areas in the occipital cortex that are more motion selective. So they respond to direction of motion or the motion of the objects. And people have shown in studies like this, where you have correlated movement. So you have a bunch of dots. And again, this coloring is just for visualization. They're not actually colored in the experiment. In the experiment, you have these dots that are moving. And maybe 10% of them are moving in the same direction. and the subjects, the monkey in this case, has to tell you which direction they're moving. And they usually have like these dots around the screen, and the monkey looks at the dot that shows the direction of the movement. So motion detection tasks like this, in a normal circumstances, a monkey would be able to do that with like 1-2% correlation. So if like even the 2% of the dots are moving together, you kind of can tell which direction that is if the rest of them are moving randomly. And that is, people argue, that is very dependent on the area, these areas of the brain, the areas MT and PO, because they saw that when there's a lesion to area MT, the motion detection threshold, the percentage of dots that have to move in the same direction for the monkey to be able to actually detect that is much higher. So you're actually making it harder for the monkey to detect motion. So people say, well, the area MT is responsible for detecting the direction of motion based on those bad results. Now, again, keep in mind, it's not that they're not able to do that anymore. Like, brain is large, and there's a lot of so-called degeneracy in the brain where different areas do same things. So if one area is down, other area can pick up. There are a lot of motion detection signal in the brain, and when you knock out one area, either permanently or with some, we talked about temporary like lesioning, like you just inject it with some liquid or something that makes it, dampen the activity. You see some changes in behavior. You don't see them completely knocked out. But this shows at least that area MT is involved in motion processing. Damage to MT results in impaired motion perception. In humans, there's a condition called motion blindness or akinetopsia, which is you don't perceive motion as motion, you perceive it as disjoint events in the world. So if a car is coming toward you, you see it at one instance, you see it another instant, you see it another instance. You don't really perceive it as motion. So area MT is important for our perception of motion. And the condition is called akinetopsia. So this was in the occipital cortex. So we talked about how V1, V2 are orientation selective. V4 is kind of color selective, but also does more high-level processing of edges and whatnot. MT and PO are kind of involved in motion processing. Again, this is not their only job, and it's just, just to give you some idea of what different areas are doing. Now, and you already know, we talked about the damage to the parietal cortex, right? What was the damage we talked about? Well, we said when people have damage to the parietal cortex, they show symptoms of neglect. Now, if the damage is only to one side, you show hemi-neglect. So the damage for this patient was to the right parietal cortex. So the person was completely neglecting the visual world on the left side. It wasn't that they were blind to it. It was that they just didn't know that existed. Like, they didn't think something was missing. And that's the quality of neglect. It's not blindness. It comes from the fact that they just cannot attend to it. They don't have access to that part of the world to know something is missing. Now, the area that, so this would be so what pathway is the one up here? What is this pathway? Dorsal. This is the dorsal pathway because it goes from, and it's shown here in the blue, it goes from the occipital cortex to, I don't know why these are in color coded. This is from the book, but it goes, this blue arrow that goes from the occipital cortex to the parietal cortex is basically going from this yellow area to this green area. Um, and this, you know, reddish or whatever color you see up there, um, that goes from the occipital cortex to the temporal area, um, is is the pathway down here. Again, these are all connected too, but we're simplifying things. Um, the area at the end, the most important area in this pathway in a way, is the inferior temporal cortex, or IT for short. The initials are IT. This area, as we said, this pathway is for recognition. It's for complex pattern recognition. For example, if we showed that in the early visual areas, cells are responsive to certain orientation of bar, in this area, cells are responsive to complex patterns of visual input. Now, this is an alternative way of showing firing rate. Instead of showing it as small, basically vertical lines that show action potential, you could show what's called firing rate as a histogram. You could say, okay, how many action potential in 100 milliseconds? How many action potential in the next 100 milliseconds? And this is histogram shows that. Does that make sense to everyone? How this is basically showing the same information, but instead of showing it as a so-called raster plot, you're showing it as a histogram. You're just showing how many action potential were in each small window. In this case, there were, let's say, I don't know, 20 action potential, 10 action potential, 5 action potential in each window. So you could see that this cell is responsive to what? Well, this cell is kind of complex, right? It's not as simple anymore. It looks like it's selective to a long vertical that is connected to a variable shape that is not like, so it has to be, you see what I mean? Like it becomes more and more complex. That's the idea, that you're not responding just to a certain orientation or direction of motion or just a simple edge, but it's a complex shape. And this cell is tuned for this or selective for this, means that this is what makes this cell really fire. So this is being contrasted here with moving edges and angles that we talked about in the earlier visual areas. In IT, sensitivity is to complex objects. So, and the organization of V1, if you remember, we said each column, each mini column here is responsive to a certain orientation. People have shown that in the IT, there's more of a shape constancy in terms of when you go down the depth of a cortical column, and a depth of a cortical column is around few millimeters. That's what that means. If you go through this few millimeters of the surface of the cortex, you could see that they're responsive to similar shapes. It's not, the effect is not as strong as the one people have found in V1, where you have selectivity to a certain orientation, but people have found some evidence. Any questions about this? Okay, so another thing about the IT cells is that the idea of retinotopy, while it makes sense in V1, right? Because V1 is a tissue and you want to be able to process the entirety of the visual field. So you have retinotopy. So every area is assigned to a certain, every tissue is processing a certain area in the visual field. When you go higher in this, when you go here, when you go higher and higher, the receptive fields become larger. They become larger and larger because you have more and more convergence, right? Earlier on, let's say from the fovea, you have some ganglion cells, they connect to LGN, the LGN to V1. The receptive fields are small. The receptive fields of a V1 cell is like a small area. When you go to V2, it becomes bigger. When you go to V4, it becomes bigger. When you reach IT, the inferior temporal, it really becomes almost 1/3 or half of the visual field. So if a cell in the IT is responding to anything in this half of the visual field, so the receptive fields become much and much longer. And the receptive field, if you remember, is the area in the visual field that this cell would respond to. So the receptive fields become larger. And this scale becomes invariant because the receptive field is so large, right? A cell that responds to a small, like a face here or a big face, it would respond to both of them because like the receptive field is larger and the cell is not as sensitive anymore to the size or the location of the object. It's rather becoming sensitive to the features of the object, right? So meaning that if a cell is trying to detect the face, you want it to be able to detect the face if it's a small or detect the same face if it's really large, right? You don't want it to be sensitive to the location or the size of the object. You want it to be only sensitive to the identity of the object. Does that make sense? So it is location and a scale invariant, but it's very sensitive to the identity, to the features of that object. So IT cells are sensitive to an object's spatial location and size, are insensitive to an object's spatial location and size, which means that it's location and scale invariant. So that's what invariant means. It means that changing the scale and location is not going to change the response of the cell. That's what that means. And IT receptive fields are huge, up to 1/3 of the entire visual field. And cells will respond to pattern falling anywhere within these large receptive fields. Now, the cells in infratemporal cortex, as I said, they're complex. But what people showed that they could be as complex as a face, right? So when they're looking at the specific face selectivity, for example, for this monkey, so this is being from the IT inferotemporal cortex of a monkey, and this is response from a specific cell. And again, we're back to this raster plot where each line is an action potential. So when there is a density of the lines, it means that this cell is really responding strongly. You could see that depending on how face-like the image is and it's whether it has all its features, the cell is responding stronger and stronger. Right? This was an important discovery that you could actually see face selectivity at a single cell level. That you could find cells that are selective to faces. Okay, this is an important thing, right? It didn't have to be that way, but you find out that through these layering of information, you could build more and more specific selectivity to a point that one cell is only responsive to a face. Now, if you've heard the fusiform face area, right? That's that's an area, a patch in the human brain in the IT, the inferior temporal cortex, in the temporal lobe as part of the ventral pathway that has very face selective cells. It responds only, so a lot of the cells in that area are selective to faces. And we call that the fusiform face area. When there is a damage to that area, so we'll talk about it in the next slide, but the damage to the IT can cause different types of agnosia, depending on the degree of damage, depending on the level of the damage, right? So earlier, I talked about visual form agnosia earlier in the slides. And I told you that that patient was not even able to copy images, right? And what did I call that? Remember? You're close. Give it a shot.
No, I
Oh, okay.
not
No,
Is it apperceptive?
apperceptive. So we talked about there's a type of agnosia that is apperceptive, where you are not able to even copy the image. So it's like a perceptual problem. Like you could look at an image, not recognize it, but also not be able to copy it. There's another form of agnosia, which is associative agnosia, meaning that when you look at a figure like this, you can actually copy it to a good degree. But if you are asked to name it, you're not going to be able to name it. This is slightly different types of agnosia. It's both of them are visual form agnosia, but one of them is kind of more severe, where you're not even able to copy it. And this is called, as I said, associative agnosia, and it's part of visual form agnosia. Now, specifically, when people have, yeah.
When you say we cannot tell you what the patterns are, it's like rather than can you describe what you're drawing? Like which direction not
You could,
meaning? What does that mean?
that means that looking at this, they don't know that it's a pig. Like they cannot,
That's like, they don't know that they are drawing.
yeah.
Okay.
So it means that they look at that and they don't recognize it as an object. Like, so to them, it's just a line pattern that they could follow, but they're not able to name it. Yeah. So when we talk about, so the area I teach of recognizing something requires you for you to, most of the time when we talk about recognizing, it's like a naming task. So you wanna name this thing as being this or that. What I had up there with this patient, for example, was that, well, even looking at this, not only they cannot tell you what it is, they cannot really copy it either. Like they're not able to perceive a form, a shape out of this object in order to copy it. Yeah, it's hard to, the thing about these cases is that it's hard to imagine what it's like, right? It's hard to imagine what it's like that a person could draw an apple from memory, but when they look at this, they're not able to do that. They're not able to recognize that's an apple. Just naming it, just that this is an apple, because they know what an apple is, right? Because when you ask them to draw from memory, they could draw this. If you tell them, okay, draw an apple, they could draw it from memory. But when you show them a new apple or a drawing of an apple and say what it is, they're not able to do it. So that's this is the app. So yeah, so naming it is a visual form agnosia, but I was saying there are degrees to it. There's not just one type, right? The lesion studies could come in different flavors. In this case, it's they call them apperceptive agnosia, where people cannot even copy it. Not only they cannot name it, that's visual form agnosia, but also they cannot copy it. In the other example that we had, this person can copy the figures, but they cannot say that this is a pig that I'm drawing, or a bear that I'm drawing. And they call them associative agnosia because it's a matter of associating things to each other, like a figure to a name or something like that. Yeah, so recognition, as I said, a lot of time is a matter of naming the stuff. So just saying what an object is. So specifically, so visual form agnosia, defining it is difficulty recognizing objects. There is a specific condition called prosopagnosia, which is the difficulty recognizing faces. And some people have that, right? They have face blindness, that they cannot. So let's say you meet them, right? They see your face, and then if they see you a few days later, they're not going to be able to recognize that you're the same person. Now, face is not the only way how we recognize objects. Like we recognize them from how we recognize people. You could recognize people from their voice. You could recognize from people from their, body movement, their gait, the way they walk. But we could use a lot of different cues in order to recognize a person. But imagine the face is not the place where it happens for these people. Like faces are not recognized. Like they don't. So why would that happen? What could that mean? Like let's think about it more conceptually. What does that mean? Is an opportunity to have a conceptual understanding of what recognition means. Okay, sure.
Firing.
Yeah.
Cells are not responding.
Yeah. So the cells are not responding in the IT, and they're damaged, right? You could induce this in animals by damaging certain, it's easy to find the fusiform face area. You could just put a monkey in a fMRI scanner and then show them pictures of faces and pictures of houses and then just subtract the two. And then it just lights up really nicely in the monkey brain where the areas are. And there are multiple patches. Fusiform face area is one of them. There are multiple patches that actually are very face selective. So if those patches are damaged, that means that these cells are not responding to faces. So if they're not responding to faces, the rest of the brain does not have access to fine-grained feature detection for faces. Does that make sense? When I say the rest of the brain, right, you have a holistic perception of an individual, right? You have a perception. A person sounds a certain way, they act a certain way, they look a certain, they smell a certain way, right? All of those things are associated with one another. Now, if you don't have, when we say a person looks a certain way, it means that you have to have features, you have to know, you have to actually process those features. You have to have depth in the way you process those features, right? The distance between their eye, right? The size of their nose or something like that, right? Those fine-grained features have to be put together to a perception of a face, right? You could actually detect maybe the size of the nose and the distance between the eyes and all the other fine-grained features, but holistically, certain cells responds to the combination of those features. The same way that certain cells respond to the combination of ganglion cell responses and some cells respond to the combination of simple cell responses, there are cells that respond to the combination of these lower level cells that respond to face patches, like different areas of the face. And those bringing them together, if those cells don't exist or they're not functioning properly, what would that cause? Well, you would not have that as part of your perception of a person because, well, you don't have that representation in the brain, right? So you have to represent someone's face in your brain in neural activity for it to be associated with all the other characteristics of the same person. And if those cells are not active, that's not part of your perception of that person. Yeah. Sure. I would say, yeah. So if you want to recognize someone, if you don't have face selective cells active, you rely on other aspects, right? As I said, you rely on the way they walk or the way they talk or something like that, right? So all those other ways, because you have cells that respond to those things, that would become part of your perception of the object. So you recognize a person as the same person that you saw 2 days earlier based on those other characteristics, because those characteristics are represented in your brain. And I can't emphasize how important that representation and language of representation is, that you need to represent things in neural activity in the brain, because brain only knows about neural activity. Now, if I want to associate a person's way of talking with the way they look, then I have to do that in my brain, and I have to do it based on neural activity of different areas. So some cells are responding to their face, some cells are responding to their voice, some cells are responding to their smell. All of those things have to be active together for me to associate all those things. And if some of them are not working, that doesn't become part of my representation of that person. Yeah? Yeah.
Is there an effective processing component to this? How someone can feel about it or the affective memories that brings up that recognition of that face effect?
I could imagine, yes. I would like to think that a lot of recognition is not impacted by those factors, but if you look at a scene and there are a lot of faces and there's one threatening face, it's likely that that would be recognized more, like in that sense, right? It gives them more advantage in that sense, but I'm not sure if your perception of an object necessarily, maybe based on the valence of your experience with them, if your perception of their other features would be impacted necessarily. But you could, yeah, you could assign negative valence to other properties of the person because of certain things. Maybe. I'm not sure. But you understand what I mean by these properties being represented in the brain at the same time for a person or for an object, for them to create one perception of it. Yeah.
What's the difference between agnosia and experience
Synesthesia?
Yeah.
Synesthesia is about one sensory modality causing another sensory modality perception. So let's say you, what's that?
No.
Yeah, so it's about, let's say you hear, some people when they hear certain music, they see a certain color. Or when they see a certain number, they see it in a certain color. So it's like you're stimulating somehow a different sensory modality based on one sensory modality. Um, but agnosia is about not being able to recognize something.
People have agnosia if they had two identical things. They would not be able to say they look the same.
Yeah, no, they cannot process that. Yeah, they cannot process that information. But if the other properties are the same, then you probably could tell that. Yeah. So that brings me to the final point, modularity. And this is another important concept where when we talk about different brain areas, when we talk about any complex system, what we strive for, what we hope for even, is modularity, right? When you think about your phone, or when you talk about a car is a modular system. You could take out the wheels, you could replace them, you could, remove the top and it would still function, right? There are a lot of modules that work together for a car to function. And any complex system, what you hope for is that divide and conquer strategy, right? If I could say, well, the eye does this, the primary visual cortex does that, the area V4 does this, and the MT does motion, and the IT does form recognition and representation, then I'm going somewhere with that, right? I'm assigning functions to these different brain areas. Each of them is a module for themselves. If I lesion this one, I take out that functionality. If I, lesion this part, I take out another functionality. That's the hope, that you could have a system that is modular enough that you could understand it by understanding its parts. And that's what people try to do with the brain. We try to assign roles to different modules. However, there are limits to this, right? Brain is a very interconnected system. It's not built of modules. Like as I said, if you look at the picture I showed about where the area of V1 is, V1 is part of the tissue, and then V2 is next to it, and then V4 is next to it, right? These are areas on the tissue that is extremely connected, like densely connected. So the arguments of modularity in the brain really hits a wall when you want to get deep at it, right? It still works. You still could say, well, they took out the hippocampus of this guy and only his long-term memory got impacted, or they took out this area like the cerebellum and only their motor movement got a little impacted. So it works on some larger scale maybe, but modularity, so I want, it's just a cautionary tale here that when we talk about modularity in the brain, it's not really extreme concept of modularity, the way a car is, the way a phone is, for example, where you could, maybe take out the battery and then place a new one. Maybe you could take out, the camera and put a new one. So that sort of modularity is nice. The brain doesn't really, fit that level of modularity. I just want you to understand when we talk about modules and when we assign function to them, these are very interconnected. And that makes the brain very complex. Because when you have such a complex system with tens of billions of units of cells, they're all doing their own thing in some way, and then they're kind of interacting with each other. And there are a lot of so-called emergent properties in this system. A whole is far more than the sum of its parts. And that's what you should, keep in mind, that the whole of the brain is so much more than just the sum of these modules. The modules are just for us to kind of understand and kind of approach the problem. But when they come together in a densely connected way, like a society, a lot of things happens where no one had predicted. They're just emerging in a society based on these somewhat, half independent, half dependent individuals that are interacting with one another. A lot of emergent properties happen, and you cannot really divide and conquer. We'll try to do it as much as we can, but keep in mind that this is just, a lot of it is on the surface and gets a little deep, but the brain has that just a different way of doing things in terms of how these parts come together and different things emerge in that complexity. So let's just always appreciate that.
Yeah, I have a question. For example, if there is a lesion in the occipital cortex,
Mhm.
that it's connected to the parietal cortex, so it could affect also the parietal cortex. And so the function is
Yeah.